Right, let's quickly go through the works of a cell in influencing what we see outside. For a cell to function, there should be cell division. And there are two types of cell division, which is mitosis and meiosis. Mitosis happens just after fertilization occurred to form a zygote, right, for the reasons of cell proliferation. And then meiosis happens during puberty stage around 11 to 15 years for the reasons of gametogenesis. All right, remember, in a cell we have a nuclear membrane, and within that nuclear membrane we have a nucleoplasm, and that nucleoplasm serves as an environment to store DNA and to facilitate its functioning. Now, inside that nucleoplasm we have a tangled network of threads known as chromatin network. Now, before cell division, which is and May, that chromatin network unwinds to form separate strands esizibiza ngoba zintoni zi-chromosomes. Furthermore, a chromosome nganye yenziwe by a strand of DNA. So never get confused when they ask you one word, the process by which a single-stranded chromosome become a double-stranded chromosome. That is DNA replication, because DNA yenza chromosomes. Moreover, chromosomes after mitosis have two sets or a double set, and we call that a diploid condition of chromosomes. And then after meiosis, they are in a single set or they have one set, and we call that a haploid condition of chromosomes. Again, chromosomes from the paternal side and the maternal side that code for the same characteristic or for a certain characteristic ziyamelana and are identical in shape and appearance, and we call those chromosomes homologous pair of chromosomes. I-DNA yethu eyenza i-chromosomes nayo yenziwe ine-building blocks or ii-monomers zayo esizibiza uba zintoni zii-nucleotides. Eza nucleotides zethu naziya, it's PSN, our phosphate, our sugar, and our nitrogenous bases. Now, nitrogenous bases zethu zine-forms ezimbini, our pu and pi, purines and pyrimidines. Right, now under our purines we've got our AG. You remember the phone, right? And then under our pyrimidines we've got RCT. You remember the shoe, right? Yeah. So this is adenine, this is guanine, this is cytosine, this is thymine. Remember, we're talking about DNA. But during what we talked about DNA replication, these nitrogenous bases, they need to form complementary bases, and that means we cross together. The adenine will stand with thymine, then cytosine will stand with guanine. Kwezi strands zezi nucleotides zethu, just a short sequence kuzo that carries a genetic code for a particular characteristic is known as a gene, and that gene basically is a portion in a DNA, right? And this gene is found in a specific position in a chromosome, and that is known as a locus. Now, i-gene yethu esiyifumana kwi-locus yayo ina two alternate forms. Sizifumana kwi-locus A1, right? But these two alternate forms provide different expressions. For example, for a gene of a height, we'll find two alternate forms of alleles, which is one for tallness and one for shortness. And the distribution of these alleles can be in three ways, which is complete dominance, incomplete dominance, and codominance. It is now these alleles that determine the genetic composition of an individual, which is the genotype. And this genotype determines the external appearance of an individual, which is the phenotype.